Hello friends, good evening. Welcome to Sam Biology class. Today we shall discuss about C-value paradox and C-value enigma. In 1948, Roger and Colette Vendrille reported a remarkable constancy in the nuclear DNA content of all cells in all individuals within a given animal species. The term C-value reflects this observed constancy. By definition, C-value is the amount of DNA in picograms contained within a haploid nucleus, for example, in a gamete. In diploid cells, it is equal to half of the total nuclear DNA present. In some cases, notably in diploid cells, the terms C-value and genome size are used interchangeably. However, in polyploids, the term C-value may represent two or more genomes. Different organisms have different amount of DNA. Some have more than others. For example, vertebrates have more DNA than invertebrates, which in turn have more than fungi. And fungi have more DNA than bacteria. Mouse and human haploid cells have 100 times more DNA than yeast haploid cells. And yeast cells have 5 times more DNA than E. coli. So the amount of nuclear DNA varies from one organism to another. One of the most notable differences between prokaryotic and eukaryotic genomes is their size. The genome of bacteria and archaea are extremely small. Their size varies from 140 kilobase pairs to about 15 megabase pairs. Most of this variability arises due to the differences in the number of protein coding genes. In contrast to this, eukaryotic genomes are larger and range more than 60,000-fold. Their size varies from 2.3 megabase pair in parasitic Microsporidium Encephalitigium intestinalis to about 150,000 megabase pair in the plant Paris japonica. Initially, it was thought that there was a correlation between the C-value of an organism and its biological complexity. If this correlation is applicable in all cases, then organisms which are considered as developmentally complex must have higher C-values than the lower organisms. But it has been found that many lower organisms have significantly larger genomes. For example, many primitive fish and amphibians have 20 times more DNA than those of human beings. In addition, the cells of some salamanders have even 40 times more DNA than those of humans. In addition to this, many morphologically similar species exhibit unexpectedly large differences in their genome size. So there is a lack of correlation between the C-value and the biological complexity of an organism. This lack of correlation is known as C-value paradox. The term C-value paradox was introduced by C.A. Thomas Jr. in 1971 while reviewing the genetic organization of chromosomes. C-value paradox refers to the lack of correlation between the genome size and the biological complexity. Initially, in order to resolve the main issue of C-value paradox, the following suggestions were made. Number 1. The paradox may arise due to inaccuracies in the estimation of the genome size, or it may be due to unusual events of genomic multiplication For example, multi-stranded mitotic chromosomes, which are seen in species with high haploid DNA content. Another suggestion was that the additional DNA may serve as a reserve for evolutionary experiment. But because natural selection is applied to the organism as it is, not as it might be, so this suggestion was considered untenable, hence ruled out. C-value paradox reflects three main issues. Number one, why do many lower organisms have significantly larger genomes than the higher organisms? Number two, why do similar species exhibit unexpectedly large difference in their genome size? And number three, why the proportion of non-coding DNA in eukaryotes is very high, even up to 90-80% in species that carry the basal C-value characteristics of the given group. The main question of C-value paradox was resolved with the discovery of non-coding DNA in eukaryotes in early 1970s. It is due to the presence of large amount of non-coding DNA, the genome size do not reflect the gene number. For example, in human being, protein coding region accounts for less than 2% of the genome size, and the remainder is various types of non-coding DNA, especially transposable elements. Now, what is C-value enigma? The term C-value enigma was coined by the Canadian biologist Dr. T. Ryan Greig. C-value enigma is an update of the more common but outdated C-value paradox. This concept is a complex puzzle which involves the following important new questions, which are the major subjects of investigation. Number one, what kinds of sequences are present in the eukaryotic genome and which one of these contribute to the extensive variability in the total genome size? Number two, how do these elements accumulate or become lost in the genome over evolutionary time scale? And number three, does this non-coding majority have any effect, positive or negative, on the organismal biology or even function for which it has been subject to natural selection at the organismal level? And number four, why some genomes remain streamlined whereas others reach a staggering size? So friends, this is all about C-value paradox and C-value enigma. For more updates, subscribe my YouTube channel, Sam Biology Class.